Hello, and welcome back to another episode of Go Beyond A&P, where we take the concepts you learn in anatomy and physiology and go a little deeper to understand not just what happens in the human body, but why. And if you've taken anatomy and physiology, you've probably learned the 5 major types of white blood cells that we see on a white blood cell differential, neutrophils, lymphocytes, monocytes, eosinophils, and basophils. And you probably had to memorize them using an acronym. Never let monkeys eat bananas. And maybe you've even had to memorize their percentages. But here's the question I want to start with. Why do we need 5 different types of white blood cells? Why doesn't the body simply make one really good immune cell that does everything? The answer is that defending the human body is far too complicated for one cell to do it all. Different threats require very different responses. So in this episode, I don't want you to memorize 5 cells. I want you to meet them. We're going to look at where they come from, why they look different, and most importantly, what each one is actually designed to do. And we're going to go beyond the traditional 5. When we get to monocytes, we'll follow them out of the bloodstream and talk about macrophages and monocyte-derived dendritic cells. When we get to lymphocytes, we'll separate them into B cells, T cells, and natural killer cells. And by the end, I want you to see these cells not as isolated entries on a laboratory report, but as specialized members of a coordinated immune system. So imagine 3 patients walk into an emergency department. The first patient has a deep cut on his hand that has become infected with bacteria. The second patient has influenza, and the virus is replicating inside his own cells. And the third patient recently traveled internationally and has acquired a parasitic infection. All 3 patients need an immune response, but should the body respond to all 3 in exactly the same way? Probably not. A bacterium growing in a wound presents a very different problem from a virus hiding inside one of your own cells, and both are completely different from a large multicellular parasite. Those patients give us reason for having different immune specialists. And we'll come back to examples like these as we meet the individual leukocytes. But before we meet those individual specialists, let's zoom out and look at the entire team, because these cells aren't working independently. They're part of a much larger network. So traditionally, we divide the immune defense into 2 broad categories: innate immunity and adaptive immunity. Innate immunity is our rapid response system. Cells such as neutrophils, monocytes, and macrophages, and natural killer cells have mechanisms that allow them to recognize broad patterns associated with infection, cellular damage, or cellular stress. Much of that recognition machinery is already encoded and ready to go, which allows these cells to respond quickly. Adaptive immunity works differently. This is where B and T lymphocytes become especially important. These cells can generate an enormous diversity of antigen receptors, allowing individual lymphocytes to recognize highly specific molecular targets. Once the appropriate lymphocyte encounters its target and receives the proper signals, it can proliferate, differentiate, and generate a highly targeted response. Some can also persist as memory cells, allowing the immune system to respond differently when that antigen is encountered again. And if you watch the episodes on vaccines or antigens, some of this should sound familiar. Vaccination takes advantage of immunological memory, and antigen recognition is fundamental to B and T cell function. But I don't want you to picture innate and adaptive immunity as 2 separate armies. They're constantly communicating. One of the most important bridges between them is antigen presentation. A dendritic cell, for example, can encounter a pathogen in peripheral tissue, capture and process its antigens, and eventually present that information to a naive T cell. We'll follow that process later in the episode. So rather than thinking of two independent immune systems, think of one interconnected leukocyte network with different cells performing different jobs. And despite all that specialization, notice what's sitting in the center of this slide, the hematopoietic source. So go back to our patient with the infected cut. The body can't afford to wait several days before responding to bacteria multiplying in that wound. Innate immune cells need to recognize the problem and begin responding rapidly. But now consider our patient with influenza. Innate immunity is still important early in that infection, but eventually we want a more targeted response against viral antigens, including virus-specific lymphocytes and potentially immunological memory. Those two aren't unrelated responses. Innate immune cells can help initiate and shape what adaptive immunity does next. That's one reason the immune response is better understood as a network than as a collection of independent cells. So despite how different a neutrophil and a T site eventually become, if we trace their ancestry back far enough, we arrive at the same place. So let's rewind the story. Before there was a neutrophil, before there was a macrophage, and before there was a B cell or a T cell, there was a stem cell in the bone marrow. So every leukocyte we're going to discuss begins with hematopoiesis, which just simply means the formation of blood cells. And at the top of this hierarchy is the hematopoietic stem cell, or HSC. And if you're coming from a traditional anatomy and physiology course, you may have learned the term hemocystoblast. That's still useful terminology, but hematopoietic stem cell is the term you'll encounter much more frequently as you move into hematology and immunology. There are two properties of the cell that I really want you to understand. The first is multipotency, meaning that it has the developmental potential to ultimately generate the different cellular components of blood. And the second is self-renewal. Some of these cells can divide while preserving the stem cell population rather than committing every daughter cell to differentiation. And that's incredibly important because your body is constantly replacing blood cells throughout your life. The hematopoietic system therefore has to solve two problems simultaneously. It has to make new blood cells while maintaining the population capable of making those cells in the future. That's why the hierarchy on this slide matters. At the top, hematopoietic stem cells have substantial self-renewal capacity and broad developmental potential. As we move downward, we encounter short-term hematopoietic stem cells and then multipotent progenitor, or MPPs. As we continue down the hierarchy, self-renewal decreases while specialization increases, eventually producing increasingly lineage-restricted progenitors. One pathway moves towards the myeloid lineage, which ultimately gives rise to cells like neutrophils, eosinophils, basophils, and monocytes. And another pathway moves towards the lymphoid lineage, which gives rise to B cells, T cells, and natural killer cells. And the real biology is more complicated than a perfectly clean family tree with rigid forks, but this hierarchy gives us an extremely useful framework. And the farther we move down the tree, the more specialized the cell becomes. So imagine a patient receives a treatment for that severely damages the bone marrow. Over time, the red blood cells begin to fall, their platelets decrease, their white blood cells decrease. At first, those might appear to be three separate problems, but they can all point us to the same place, the bone marrow. Red blood cells, called erythrocytes, Platelets, thrombocytes, and white blood cells, leukocytes, ultimately depend on hematopoiesis. Damage the system near the top of that hierarchy, and the consequences can appear across multiple blood cell populations. It helps us appreciate just how important that relatively small population of hematopoietic stem cells really is. So now we've reached the point where our descendants begin choosing very different developmental paths. One direction takes us towards the myeloid lineage. The other takes us towards the lymphoid lineage. But as we're about to see, even that apparently simple split isn't quite as clear as the traditional family tree makes it look, because dendritic cells make the story a little bit more interesting. So we've reached one of the major branching points in hematopoiesis. From the multipotent progenitor, developing blood cells become progressively committed towards different lineages, and the traditional model divides those pathways into myeloid and lymphoid progenitors. The common myeloid progenitor gives rise to the granulocytes we'll meet shortly, neutrophils, eosinophils, and basophils, as well as monocytes and other blood cell lineages. The common lymphoid progenitor leads us towards B lymphocytes, T lymphocytes, and natural killer cells. This division is useful because it helps explain why cells that eventually look and behave very differently are still developmentally related. But I want to add an important caveat. The actual process of hematopoiesis isn't quite as simple as a family tree with two perfectly defined branches. Lineage commitment occurs progressively, and some immune cell populations don't fit neatly into the traditional diagram. And dendritic cells are a particularly good example. Different dendritic cell populations can arise through developmental pathways that complicate a simple myeloid versus lymphoid classification. And monocytes can also generate monocyte-derived dendritic cells under certain conditions. So use this bifurcation as a framework, not an absolute rule. The major idea is that as progenitors move along these developmental pathways, they progressively acquire the machinery that allows them to perform very different immune functions. So why does this lineage matter clinically? So imagine a laboratory report showing an abnormal number of one particular leukocyte population. Knowing the developmental lineage gives us another way to think about what we're seeing. Is the change isolated to one mature cell population, or are multiple related populations affected? And if something disrupts an early progenitor, the consequences can extend to several of its descendants. We aren't going to go into leukemias or bone marrow disorders in this episode, but the family tree we're building now becomes extremely important when you eventually study these conditions. So understanding where a cell comes from helps you understand why disturbances at different points in development can produce very different patterns in the blood. So now that we know where these cells come from, let's switch perspectives. Instead of classifying leukocytes by ancestry, let's classify them by something much easier to see: what they actually look like under the microscope. And that's where we get two terms you've probably heard of before: granulocytes and agranulocytes. If you've ever looked at a stained blood smear, you've probably noticed that white blood cells don't all look alike. One of the traditional ways we classify them is based largely on their morphology, particularly the appearance of the cytoplasm and the nucleus. The granulocytes are neutrophils, eosinophils, and basophils, and as their name suggests, these cells contain prominent cytoplasmic granules that become visible with standard staining techniques, although the appearance of those granules differs dramatically among the three cell types. Their nuclei also tend to be lobed or segmented, while the agranulocytes are monocytes and lymphocytes. Now, the name agranulocyte can be slightly misleading because it doesn't mean these cells contain absolutely no intracellular granules. Rather, they lack the large, conspicuous, specific granules that characterize the granulocytes on a routine blood smear. Monocytes are generally larger cells with an indented or kidney-shaped or horseshoe-shaped nucleus, while many circulating lymphocytes are dominated visually by a large, round nucleus surrounded by a relatively thin rim of cytoplasm. But here's the important part. These morphological differences aren't just useful for identifying cells on an exam. Structure and function are always connected. Granules contain molecules that can be released or deployed during immune responses. Nuclear morphology can help us identify a cell's lineage, and the appearance of a leukocyte provides clues about the specialized machinery inside of it. So when you look at these 5 cells, don't just ask which one is the neutrophil. Start asking, why does the neutrophil look different from the lymphocyte? And what does that tell me about what it does? And this is exactly what happens when a clinician orders a complete blood count with a differential. A total white blood cell count tells us how many leukocytes or white blood cells are circulating, but the differential tells us how that population is distributed among neutrophils, lymphocytes, monocytes, eosinophils, and basophils. So 2 patients could have the exact same total white blood cell count and still have very different differentials. One might have a predominance of neutrophils, while another has a greater proportion of lymphocytes or eosinophils. We're deliberately not going to turn this episode into a discussion of disease-specific patterns, but this is why learning the individual cell types matter. The differential isn't simply 5 percentages printed underneath the white blood cell count. It's more of a snapshot of the different immune cell populations circulating at that moment. And now we're ready to meet our first specialist. If bacteria enter damaged tissue and the body needs a large number of cells that can divide or that can arrive quickly, engulf microbes and kill them, one leukocyte becomes particularly important. It's also the cell you're most likely to find when you look at a normal white blood cell differential, and that's the neutrophil. Neutrophils are the most abundant leukocytes in normal adult peripheral blood, typically accounting for about 50 to 70% of circulating white blood cells, and they're built for rapid innate immune defense. Their distinctive multi-lobed nucleus is why you may hear them described as polymorphonuclear leukocytes, or PMNs. But the real story is what's packed inside the cell. Neutrophils contain multiple types of granules loaded with antimicrobial molecules and enzymes, giving them an impressive arsenal that can be deployed against invading microbes. They also express pattern recognition receptors that allow them to recognize conserved molecular patterns associated with microorganisms and tissue injury. And once activated, neutrophils can migrate rapidly towards affected tissue, engulf microbes through phagocytosis, expose those microbes to antimicrobial granule contents, and generate reactive oxygen species through the respiratory burst. The enzyme complex NADPH oxidizes NADPH oxidase plays a central role in that oxidative response by helping generate reactive oxygen intermediates that contribute to microbial killing. So when I call neutrophils the rapid response force, I mean exactly that. These aren't cells designed to spend days carefully developing a highly antigen-specific response. They're designed to recognize that something is wrong, get to the problem quickly, and attack it. And because their weapons can also damage surrounding host tissue, neutrophil activity has to be powerful but controlled. So let's go back to our first patient, the person with the infected hand or the infected cut on his hand. Bacteria have crossed the physical barrier of the skin and they're multiplying under the tissue. Resident cells recognize signs of infection and release inflammatory mediators. The local endothelium changes, chemical gradients develop, and circulating neutrophils are recruited toward the affected area. And if that wound begins producing pus, much of what you're looking at reflects the aftermath of this battle. Accumulated neutrophils, cellular debris, microorganisms, and inflammatory material. So the neutrophil on this slide isn't simply the most common type of white blood cell. It's part of a rapid mobilization system that can move cells from the circulation into threatened tissue and place antimicrobial machinery directly where it is needed. But saying that neutrophils attack bacteria leaves out the most important part of the story. How does a neutrophil know where to go? How does it get out of the bloodstream? What happens after it catches a microorganism? And what does it do if simply engulfing that target isn't enough? So on the next slide, we're going to follow a neutrophil through the actual sequence of attack, from chemotaxis to phagocytosis to oxidative killing, and one of its most dramatic weapons, the neutrophil extracellular trap. So on the last slide, we met the neutrophil as our rapid response force. So let's follow one in battle. The first challenge is getting the neutrophil to the right place. And that's where chemotaxis comes in. Chemical signals generated during an infection and tissue injury create gradients that neutrophils can detect and follow towards the affected tissue. Once there, one of their major weapons is phagocytosis. The neutrophil surrounds a microorganism and encloses it inside a membrane-bound compartment called a phagosome. But capturing the organism just isn't enough. The neutrophil now has to kill it. Granules confuse with the phagosome and deliver antimicrobial surfaces, while the respiratory burst generates reactive oxygen species that create an extremely hostile intracellular environment. And then we have one of the most dramatic neutrophil defenses, neutrophil extracellular traps, or NETs, N-E-T. Neutrophils can release networks composed largely of decondensed chromatin decorated with antimicrobial proteins. Think of them almost like molecular nets cast into the extracellular environment. They can trap microbes, concentrate antimicrobial molecules around them, and potentially limit their spread. You'll sometimes hear this process called netosis, although the biology is more complicated than the original idea that every net-producing neutrophil simply dies in that process. So a neutrophil has multiple options available. Find the problem, engulf the target, attack it chemically, and under appropriate circumstances, deploy extracellular traps. And that's quite an arsenal for a cell that many students initially learn simply as the most abundant type of white blood cell. So let's return to our infected wound. So imagine bacteria are multiplying in the tissue. Neutrophils recruited from our bloodstream follow chemical gradients towards that site, encounter the bacteria, and begin engulfing them. Inside the neutrophil, antimicrobial granules and reactive oxygen species help destroy what has been captured. So if you could shrink yourself down and watch this response happening, it wouldn't look like a passive collection of white blood cells just sitting around an infection. It would look like an activated cellular battlefield. And there's an important trade-off here. The same reactive molecules, enzymes, and inflammatory mechanisms that make neutrophils effective against microorganisms can also injure surrounding tissue. That's why inflammation can protect us and damage us at the same time. The weapons don't suddenly become harmless simply because our own cells are nearby. Neutrophils are extremely effective against targets they can reach, engulf, and attack. But now let's change the scale of the problem. What happens when the target is far too large for a white blood cell to simply swallow? You can't exactly ask a neutrophil to phagocytize a parasitic worm. For that kind of challenge, we're going to need a different strategy, a different granulocyte. So now we meet the eosinophil. Eosinophils are much less abundant than neutrophils in normal peripheral blood, but their relative scarcity doesn't make them unimportant. Their appearance gives us our first clue about how specialized they are. Eosinophils typically have a bilobed nucleus and prominent cytoplasmic granules that stain strongly with eosin, giving them their characteristic reddish-orange appearance. Those granules contain potent molecules, including major basic protein, eosinophil cationic protein, eosinophil peroxidase, and eosinophil-derived neurotoxin. Rather than relying primarily on phagocytosis, eosinophils can release these granule contents into the extracellular environment, which becomes particularly useful when confronting targets too large to engulf. This is one reason eosinophils are traditionally associated with defense against multicellular parasites such as helminths. But I don't want you to walk away thinking that eosinophils are simply the parasite cell. They also participate in type 2 immune responses and communicate with other components of the immune system through cytokines and other mediators. Their activity can therefore contribute to a much broader immune environment. So compared with our neutrophil, we're already seeing specialization in action. The neutrophil is extraordinarily effective, rapid phagocyte. The eosinophil brings a different toolkit, one particularly suited to extracellular targets where releasing toxic molecules onto the target can be more useful than trying to engulf it. So now let's return to our third patient, the traveler with a parasitic infection. Imagine the immune system encounters a helminth. This is not a bacterium a neutrophil can simply surround and place inside a phagosome. The parasite may be vastly larger than any individual leukocyte, so the strategy changes. Instead of asking, how do I swallow this, the immune system can recruit cells capable of attacking the target from the outside. Eosinophils can participate by releasing toxic granule proteins onto the parasite's surface as part of a coordinated type 2 immune response. And this illustrates something important about immune specialization. The best defensive strategy depends partly on the physical nature of the threat. What works beautifully against a bacteria may be completely impractical against a multicellular parasite. So far, our granulocytes have been relatively easy to understand as effector cells. Neutrophils can rapidly attack microbes, and eosinophils can help confront large extracellular targets. Our third granulocyte is different. It's rare, extremely rare in the bloodstream, and its most important contribution isn't necessarily what it directly kills. Instead, think of this next cell as more of an alarm system capable of rapidly changing the environment around it. So basophils are the least abundant of our 5 major circulating leukocyte categories, typically accounting for less than 1% of peripheral white blood cells. But this is a great example of why abundance doesn't necessarily tell you importance. Basophils contain prominent cytoplasmic granules, and those granules can be so dark with routine staining that they particularly obscure or partially obscure the nucleus. Functionally, basophils are particularly associated with type 2 immune responses and express high affinity receptors for IgG called Fc epsilon Ri receptors. When appropriately activated, they can rapidly release preformed mediators or preformed mediators such as histamine and generate additional lipid mediators and cytokines. Histamine can influence vascular permeability and blood flow, while cytokines such as interleukin 4 and interleukin 13 can help shape the surrounding immune response. So instead of thinking of basophils simply as the histamine cell, think of it as a rare but potent signaling cell capable of rapidly modifying the local inflammatory environment. And here's an important distinction. Basophils and mast cells share several functional characteristics, including high affinity IgE receptors and the ability to release histamine. But they are not the same cell. Basophils are circulating granulocytes produced through hematopoiesis, whereas mast cells are distinct tissue-resident immune cells within their own developmental biology. They may use some of the same weapons, but they aren't interchangeable names for the same population. So think about what happens after a mosquito bite. Within a short period of time, the area becomes red, warm, swollen, and itchy. Histamine is one of the mediators that can contribute to those vascular and sensory changes. In a real mosquito bite, tissue-resident mast cells are particularly important. So I don't want to incorrectly attribute that reaction entirely to basophils, but it's a useful illustration of what histamine can do to a local tissue environment. Basophils use some of that molecular machinery and can participate in IgE-associated and type 2 immune responses. The broader lesson is that an immune cell doesn't have to engulf or directly kill something to make an enormous difference. Sometimes changing blood vessels, recruiting other cells, and altering the cytokine environment itself is a powerful immune function. So we've met the 3 granulocytes: neutrophils, eosinophils, and basophils. Each carries visible granules, but those granules contain very different tools because these cells have very different jobs. Now we're leaving the granulocytes and moving to a cell with an unusual identity. In the bloodstream, we call it a monocyte. But for many monocytes, circulation isn't the final destination. Once recruited into tissue, these cells can change dramatically. And that's where the next part of our story begins. So now we move from the granulocytes to the largest of the 5 major leukocytes normally found in peripheral blood, the monocyte. Monocytes typically account for about 2% to 8% of circulating white blood cells, and morphologically, they're fairly easy to recognize. They're large cells with abundant cytoplasm and a nucleus that is often described as kidney-shaped or horseshoe-shaped. But what makes monocytes especially interesting is that the bloodstream is often more of a highway than a final destination. In response to appropriate signals, monocytes can leave the bone marrow, circulate through the blood, interact with vascular endothelium, and eventually migrate into tissues. The sequence shown on the slide, rolling, adhesion, crawling, and transmigration, illustrates that leaving the circulation is an active, highly regulated process. And not all circulating monocytes are identical. In humans, we generally recognize classical monocytes, which are CD14 positive, CD18 negative, intermediate monocytes, which are CD14 positive, CD18 positive, and classical and non-classical monocytes, which are CD14 positive and CD18 2 positive. Classical monocytes represent the largest population and are particularly important in recruitment to tissues, while non-classical monocytes are often described as vascular patrollers because they crawl along the luminal surface of blood cells and survey the endothelium. And once recruited into tissues, monocytes can undergo substantial functional changes and, depending on the signals that they receive, can differentiate into cells including macrophages or monocyte-derived dendritic cells. So when you see a monocyte on a blood smear, you're looking at a circulating immune cell whose identity and function may change considerably after it reaches its destination. So imagine you scrape your knee and introduce bacteria into the damaged tissue. Neutrophils may dominate the earliest wave of recruited leukocytes, but the immune system doesn't end there. Circulating classical monocytes can also be recruited towards inflammatory signals, interact with activated vascular endothelium, leave the bloodstream, and enter the affected tissue. Once there, the environment begins influencing what those cells become and what they do. Some can differentiate into macrophages that phagocytose microorganisms and cellular debris, release signaling molecules, and participate in coordinating the response. So when we draw a monocyte in the bloodstream and a macrophage in the tissue, we're not necessarily looking at two completely unrelated cells. In many inflammatory settings, we're looking at different stages in the life of a recruited myeloid cell. And this raises an obvious question. If a monocyte can enter a tissue and become a macrophage, does that mean every macrophage in your body started out as a circulating monocyte? That's the traditional A&P story, but the real answer is no. And the reason why takes us into one of the most interesting changes in our understanding of the immune system. The word macrophage literally means big eater, which is a pretty good description of one of its major abilities. Macrophages are highly effective phagocytes that can engulf microorganisms, damaged cells, cellular debris, and other materials in tissue. But calling them big eaters dramatically undersells what they do. Macrophages can also act as tissue sentinels, produce cytokines, interact with other immune cells, participate in tissue maintenance, and can contribute to both inflammatory responses and their resolution. And here's the Go Beyond A&P part of the story. Not every tissue macrophage came from a monocyte circulating in your blood yesterday. Some macrophage populations can be replenished from recruited monocytes, particularly during inflammation, but many tissue-resident macrophages are established much earlier in development from embryonic progenitors and can maintain themselves locally through self-renewal. Different tissues give rise or even give their resident macrophages specialized identity, reflecting the environment in which they live. The other important idea on the slide is the spectrum or the activation spectrum. You may have learned about M1 and M2 macrophages as they were two completely separate macrophage types. And that's a useful introductory model, but the real macrophage biology is much more fluid. The M1-like end of the spectrum describes macrophages with more strongly pro-inflammatory and antimicrobial characteristics, while the M2-like end emphasizes functions associated with resolution, modeling, and tissue repair. In the body, macrophages can occupy many functional states between those endpoints and can change their behavior as the signals in their environment change. So macrophages aren't simply one cell with one job. They're adaptable tissue cells whose origin, location, and local environment help determine what they do. So think about what happens after you cut your skin. Early in the response, the priority is defense, contain microorganisms, clear the damaged cells, and generate inflammation. But eventually, the goal has to change. If the immune system remains permanently locked into an aggressive inflammatory state, the tissue can't return to normal. Debris has to be removed, inflammation has to resolve, and tissue repair has to occur. Macrophages can participate across those changing phases. The macrophage helping create an antimicrobial inflammatory environment early in an injury doesn't have to behave identically throughout the entire healing process. And as the molecular signals change around it, its functional program can change as well. That's why thinking in terms of an activation spectrum is more useful than imagining that every macrophage is permanently stamped as M1 or M2. So macrophages show us that immune cells can do much more than destroy invaders. They can sense what's happening in a tissue and communicate with other cells. But now we're going to meet a cell that takes that communication role to another level. Imagine a cell that encounters something suspicious in your skin, collects molecular evidence, travels to a lymph node, and then presents that evidence to T lymphocytes. That's the remarkable job of the dendritic cell. Dendritic cells are among the immune system's most important professional antigen presenting cells, or professional APCs. And their defining role is to connect information gathered in peripheral tissues with the activation of adaptive immune responses. Their name comes from their branching dendrite-like cellular processes, which increase the surface area available for interaction with their environment and other cells. So dendritic cell biology is more complex than a single cell type, and the slide introduces several important populations, including conventional dendritic cells, CDC1 and CDC2, and plasmacytoid dendritic cells, or PDCs. Conventional dendritic cells arise through a dedicated dendritic cell developmental pathway, while monocytes can also generate monocyte-derived dendritic cells under inflammatory conditions. That's why I don't want you to simply memorize monocytes become dendritic cells as a universal rule. Functionally, dendritic cells act as sentinels. In peripheral tissues, they sample their environment and capture antigens. And when appropriately activated, they undergo a remarkable transformation. Antigen processing and presentation functions become increasingly important. Expression of molecules involved in T cell activation increases, and the dendritic cell can migrate towards secondary lymphoid tissues. There, it can display processed antigen fragments on MHC molecules to T lymphocytes. If you've watched the episode on antigens, this is where that molecular story becomes a cellular one. The antigen isn't useful to a naive T cell simply because it exists somewhere in our body. A dendritic cell can capture that information, process it, carry it to the appropriate location, and present it in a form that a T cell can recognize. That's why I like to think of dendritic cells as professional messengers. They don't simply detect the problem, they help tell the adaptive immune system what the problem looks like. So imagine you receive a vaccine in your upper arm, your deltoid muscle. The material doesn't need to somehow travel around the body personally, introducing itself to every T lymphocyte. Instead, antigen presenting cells in the local environment can encounter vaccine associated antigen and inflammatory signals, process that antigen, and help carry information toward draining lymph nodes. Dendritic cells are important, particularly important, because they are exceptionally capable of activating naive T cells. So that sore arm after vaccination is only the visible location where the story begins. And at the cellular level, information gathered in the tissue can be transported to a lymphoid organ where an adaptive immune response begins to take shape. This is one of the clearest examples of the innate and adaptive immune systems functioning as an interconnected network that we saw back on slide 2. But there's still a missing piece. Capturing antigen and carrying it to a lymph node doesn't automatically activate a T cell. The dendritic cell has to deliver the right information in the right way. And it turns out that simply showing a T cell on an antigen is not enough. On the next slide, we're going to follow the dendritic cell from antigen capture to T cell activation and see why activating a naive T cell requires not one signal, but a coordinated set of signals. So on the last slide, we introduced dendritic cells as professional messengers. Now let's follow the message. The process begins in peripheral tissue, where an immature dendritic cell captures antigens through mechanisms such as phagocytosis or receptor-mediated endocytosis. But capturing the antigen is only the beginning. The dendritic cell processes that material into smaller fragments, migrates through the lymphatic system toward a draining lymph node, and undergoes maturation along the way. Once there, it can interact with naive T lymphocytes. And here's the critical point. Activating a naive T cell requires more than simply displaying an antigen. Think of it as a three-part conversion. Signal 1 provides specificity. A peptide is displayed on an MHC molecule and recognized by the T cell receptor. That tells the T cell this is an antigen, but signal 1 alone is not enough. Signal 2 provides co-stimulation through molecules such as CD80 or CD86 on the dendritic cell interacting with CD28 on the T cell. And you can think of this as confirmation that the antigen is being presented in an appropriate activating context. Then signal 3 comes from cytokines produced in the surrounding environment And these signals help influence what kind of functional response the activated T cell develops. So the dendritic cells isn't simply holding up a molecular photograph and saying, "Have you seen this?" It's providing antigen-specific information, contextual confirmation, and instructions that help shape what happens next. That's why dendritic cells form such an important bridge between innate detection and adaptive immunity. So let's continue with our vaccination example. Antigen from the vaccine is captured in the tissue, but imagine what would happen if any random antigen encounter automatically triggered a massive T cell response. We encounter enormous numbers of harmless molecules every day. The immune system needs context. Dendritic cell maturation helps provide that context. When antigen capture occurs alongside signals indicating inflammation or danger, then dendritic cell can increase co-stimulatory molecules, migrate towards a lymph node, and become more effective at activating naive T cells. So the success of an immune response doesn't necessarily or solely depend on whether an antigen is present. It also depends on how that antigen is encountered and what additional signals accompany it. That's one reason vaccines are designed not merely to expose the immune system to an antigen, but to generate the appropriate immunological context for productive adaptive responses. At this point, the dendritic cell has done its job. It captured information in the tissue, carried that information to a lymphoid organ, and presented it to the adaptive immune system. So now it's time to meet the cells waiting on the other side of that conversation, the lymphocytes. When you look at a standard white blood cell differential, you see one category labeled lymphocytes, but that single word hides an extraordinary amount of biological diversity. Three major populations we'll focus on are B lymphocytes, T lymphocytes, and natural killer cells. B cells and T cells are central components of adaptive immunity, while NK cells, or natural killer cells, are lymphocytes that function primarily as part of the innate immunity. So even the word lymphocyte doesn't automatically mean adaptive immune cell. What makes B cells and T cells particularly remarkable is their receptor diversity. Each developing B lymphocyte or T lymphocyte generates its own antigen receptor specificity through genetic rearrangement. The result is an enormous population of lymphocytes carrying different receptors and therefore capable of recognizing different molecular targets. Before an antigen ever even enters your body, somewhere within that diverse population, there may be already a small number of lymphocytes with receptors capable of recognizing it. When the appropriate lymphocyte encounters its antigen and receives the necessary activation signals, that cell can undergo clonal expansion. In other words, one rare cell with the appropriate receptor can proliferate into a large population of cells carrying that same antigen specificity. Some become effector cells that participate in the current response, while others can become long-lived memory cells. That's one of the defining features of adaptive immunity: specificity combined with the ability to remember. And this is why the lymphocyte shown on a routine blood smear is deceptive. Under the microscope, two lymphocytes may look almost identical, but molecularly, their antigen receptors may allow them to recognize completely different targets. So imagine you've never encountered a particular virus before. At the beginning of the infection, the virus isn't greeted by millions of lymphocytes already dedicated to that exact viral antigen. Instead, among the enormous diversity of B cells and T cells that live in your body, there may be a relatively rare clones whose receptors happen to recognize relevant viral antigens. Once those cells receive the appropriate signals, they proliferate. One becomes thousands, and thousands become millions of antigen-specific descendants. After the infection resolves, some of those cells may persist as memory populations. If the same antigen is encountered again, the immune system is no longer starting from the same baseline. And that's the biological principle underlying immunological memory, and one of the major reasons vaccination can change how the body responds to a future encounter. But B cells and T cells don't recognize antigen in the same way, and they don't solve the problem the same. So let's separate our adaptive arsenal. We'll begin with the lymphocyte responsible for one of the most recognizable products of the immune system, which is the antibody. B lymphocytes develop and mature in the bone marrow, and each naive B cell displays memory-bound immunoglobulin that functions as B cell receptors, or BCRs. One of the most important distinctions between B cells and T cells is how they recognize an antigen. A B cell receptor can bind directly to an appropriate antigen in its native form. The antigen doesn't necessarily have to be presented to the B cell on an MHC molecule first. When a B cell encounters an antigen that matches its receptor and then receives the additional necessary signals for activation, often including help from CD4 positive T cells, the B cell can undergo clonal expansion and differentiation. Some descendants become plasma cells, which are essentially antibody-producing factors capable of secreting large quantities of immunoglobulin. Those antibodies can circulate through body fluids, bind specific antigens, neutralize certain targets, promote opsonization, and participate in other immune effector mechanisms. Other activated B cells become memory B cells, which can persist long after the original immune response has resolved. So when we talk about humoral immunity, we're talking about immunity mediated substantially through soluble molecules and body fluids, particularly antibodies. But notice the transformation that occurs. The naive B cell begins as a cell displaying its antigen receptor on its own membrane. After activation, some of its descendants become plasma cells whose job primarily is to manufacture and secrete enormous quantities of soluble versions of that antigen recognition molecule. The specificity hasn't been abandoned. We've taken that highly specific recognition system and turned it into a secreted weapon that can operate throughout the extracellular environment. So think about what happens after a successful vaccination. Initially, antigen-specific B cells may be extremely rare, but once an appropriate B cell is activated, clonal expansion can generate descendants that differentiate into antibody-secreting plasma cells and memory B cells. Some plasma cell populations can continue contributing antibodies over time, while memory B cells provide a reservoir capable of responding to future antigen exposure. And this is why measuring antibodies can sometimes give us evidence of previous antigen exposure or vaccination. But it is also important to remember that antibodies are only one part of adaptive immunity. A person can have meaningful immunological memory that isn't completely described by a single antibody concentration. because T cells, memory B cells, and long-lived plasma cells are all parts of the larger adaptive response. B cells solve the antigen recognition problem by producing antibodies that can operate in the extracellular environment. But what if the threat is inside one of your own cells? An antibody can't simply enter every cell looking for a virus. For that, the immune system needs another strategy. Instead of recognizing free antigen, our next lymphocyte specializes in examining pieces of antigen displayed on the surfaces of cells. And before it can do it safely, it has to go through one of the most rigorous training programs in the immune system. So next we're heading into the thymus to meet the T lymphocyte. So now we move to T lymphocytes, and one of the first things that distinguish T cells from B cells is where they complete their development. T cell precursors originate from hematopoietic cells in the bone marrow, but immature T cells travel to the thymus, and that's where an extraordinary selection process occurs. Developing T cells generate unique T cell receptors, or TCRs, but simply producing a receptor isn't enough. That receptor has to be useful without being dangerously self-reactive. So developing T cells essentially have two major tests to pass. During positive selection, cells must demonstrate that their T cell receptors can appropriately recognize self-MHC molecules. And if they can't adequately interact with MHC, they won't be useful for normal T cell function and are eliminated. Then comes negative selection, where T cells that react too strongly to make or to self antigens are preferentially removed, helping promote central tolerance. These cells that successfully make it through this process can mature into populations that include CD4 positive helper T cells and CD8 positive cytotoxic T cells. CD4 T cells help coordinate immune responses through cytokines and interactions with other immune cells, while CD8 T cells can recognize and kill certain infected or abnormal host cells. And this brings us back to our antigen episode. Unlike B cells, T cells generally don't recognize free, intact antigen directly. So T cell immunity gives us a very different solution to antigen recognition, one that allows the immune system to inspect information being displayed by our own cells and by professional antigen presenting cells. Let's return to our patient with influenza. The virus has a major advantage. It can enter the patient's own cells and use their machinery to replicate. Antibodies can be useful and extremely useful against viral particles while they're extracellular, but once the virus is inside the host cell, the immune system needs another way to detect the problem. Infected cells can display viral peptides on MHC class I molecules. A CD8 positive T cell with the appropriate receptor can recognize that peptide MHC combination and, once properly activated, kill the infected cell. That may sound drastic, but from the immune system's perspective, sacrificing one infected host cell can help eliminate a factory producing thousands of new viral particles. Meanwhile, the CD4 positive T cells can help coordinate other components of the immature response. So our influenza patient demonstrates why antibodies and T cells aren't redundant. They're solving different parts of the same problem. But here's an interesting problem. What happens if an abnormal cell stops displaying enough MHC class I? A CD8 T cell depends on antigen being presenting in the context of MHC class I, so reducing that signal might seem like a clever way for an infected or abnormal cell to hide. Unfortunately for that cell, the immune system has another lymphocyte with a very different set of rules. It doesn't need a rearranged antigen-specific T cell receptor, and sometimes the absence of a normal signal is exactly what gets its attention. So now we're going to talk about the natural killer cell. Natural killer cells, or NK cells, gives us a fascinating combination of characteristics. Morphologically, they're lymphocytes, and developmentally, they arise from the lymphoid lineage. But functionally, they're major components of the innate immunity. So unlike B cells and T cells, NK cells don't rely on rearranged antigen-specific B cell or T cell receptors to decide whether to attack. Instead, their behavior reflects the balance between activating and inhibitory signals received from the cells they encounter. One of the most important inhibitory signals involves MHC class I. Normal nucleated cells generally express MHC class I, and when an NK cell inhibitory receptor recognizes that signal, it essentially tells the NK cell, this cell doesn't belong here, or this cell belongs here, don't kill it. But some virus-infected or transformed cells can reduce MHC class I expression. Now that inhibitory signal becomes a weaker or disappears, a concept often called missing self-recognition. If activating cells are sufficiently strong and inhibitory signals are insufficient, the balance can shift towards killing. Once activated, NK cells can release perforin, which helps create pores in the target cell membrane, and granzymes, which enter the target and help initiate apoptosis. NK cells can also produce cytokines such as interferon gamma, which allows them to influence other components of the immune response. So the NK cells operate almost like a biological logic gate. Activating signals tell it that there may be a problem. Inhibitory signals tell it to leave a healthy cell alone, and the balance between those inputs help determine whether the target survives. So imagine a cell becoming infected with a virus that interferes with MHC class I expression. From the virus's perspective, that may be advantageous because reduced MHC class I can make it harder for CD8 T cells to recognize viral peptides displayed by the infected cell. But now the virus has created a different problem. The reduction in MHC class I removes an important inhibitory signal to NK cells. In other words, hiding from one arm of immunity can make the infected cell more suspicious to another. NK cells don't simply attack every cell with reduced MHC class I because activating and inhibitory signals still have to be integrated. But this gives us a beautiful example of redundancy in immune defense. The body doesn't rely on one recognition strategy. Different immune cells can detect different abnormalities in fundamentally different ways. And now we've met our major players. neutrophils, eosinophils, basophils, monocytes, macrophages, dendritic cells, B cells, T cells, and NK cells all have different developmental histories and different weapons. So let's return to the question we started with. Why doesn't the body simply make one good immune cell that does everything? So we started this episode with the 5 familiar names you see on a white blood cell differential, but now those names should mean something more than 5 percentages on a lab report. We've traced them back to common hematopoietic stem cell and watched the developmental potential gradually give way to specialization. We saw neutrophils become rapid against phagocytic and antimicrobial responders. Eosinophils deploy granule contents against large extracellular targets. Basophils act as potent inflammatory signalers, and monocytes leave the circulation and contribute to a specialized tissue populations. We saw macrophages function as adaptable tissue sentinels, and dendritic cells carry information from peripheral tissues to the adaptive immune system. Then we opened up that deceptively simple category called lymphocytes and found B cells capable of producing antibody and memory, T cells capable of coordinating responses or killing specific target cells, and NK cells capable of recognizing abnormal cells through an entirely different balance of activating and inhibitory signals. That's why the leukocyte differential shown on this slide is useful but incomplete as a picture of immunity. These cells don't function in isolation. Their arsenals are complementary, and their continuous crosstalk allows the immune system to accomplish things that no single leukocyte should accomplish or could accomplish alone. A neutrophil doesn't need to become a B cell. A B cell doesn't need to behave like an NK cell. Specialization is the point. The immune system works not because one cell can do everything, but because different cells can do different things extraordinarily well and communicate well with one another while doing them. So let's go back to the 3 patients we met at the beginning. The patient with the infected wound can recruit rapid innate defenses, including neutrophils and monocytes, while macrophages and other cells help coordinate what happens in the tissue. The patient with influenza needs early innate defenses, but also benefits from highly specific adaptive responses involving B cells, antibodies, CD4 T cells, and CD8 T cells, while NK cells provide another strategy for recognizing certain abnormal host cells. And our patient with the parasitic infection presents a completely different physical challenge, one in which eosinophils and type 2 immune responses can become particularly important. None of these responses belongs just to one leukocyte, and that's the important part. The immune system combines different cells, receptors, signals, and effector mechanisms according to the type of challenge it encounters. So 3 patients didn't simply need more white blood cells. They needed the right immune cells performing the right functions at the right time. And that's where I want to leave you. So the next time you see neutrophils, lymphocytes, monocytes, eosinophils, and basophils listed on the C. diff report, or not C. diff, CBC diff differential, don't just see 5 numbers. Think about the family tree behind them, the specialized machinery inside of them, and the enormous network of communication connecting them. Because a white blood cell differential may fit on a few lines of a laboratory report, but behind those numbers is an entire immune system We've covered a lot of ground in this episode. We started with a single hematopoietic stem cell and followed its descendants as they became increasingly specialized with members of the immune system. And along the way, we encountered the 5 types of white blood cells, but we also discovered that those 5 categories only begin to describe the complexity of leukocyte biology. We followed monocytes into the tissues, explored macrophages and dendritic cells, and separated lymphocytes into B cells, T cells, and natural killer cells. More importantly, I hope you leave this episode understanding that the immune system isn't simply a collection of cells that attack things. It's a coordinated network built around specialization, communication, and cooperation. So there really isn't a separate clinical connection for this slide because the clinical connection has been the story we followed throughout this entire episode. The infected wound, the viral infection, the parasitic infection, and the vaccine response all showed us the same functional or fundamental principle from different perspectives. An effective immune response depends on matching the right cellular tools to the biological problem. A CBC may report leukocytes as numbers and percentages, but every one of those numbers represents a population of cells with its own developmental history, recognition mechanisms, and signaling pathways, as well as specialized functions. So the references shown here provided the scientific foundation for this episode and are a great place to start if you want to explore leukocyte development and function in greater detail. So thank you for joining me for this episode of Go Beyond A&P. As always, learn it for the exam, understand it for life, and until next time, stay curious and keep learning.